Hello everyone. Today we are going to talk about oxidative phosphorylation. Last time we talked about electron transport, and today we want to talk about how it's linked to the synthesis of ATP. But before we do that, I wanted to talk about how the electron transport chain was worked out. It was a very difficult task to do this. One of the ways that you can do this is that you can have cells in culture and you can add various inhibitors. So there are inhibitors, rotenone and amytal will block electron flow between complex 1 and coenzyme Q. If you added antimycin A to your culture, that would block electron flow from QH2 to cytochrome C1. And finally, if you added either cyanide, azide, or carbon monoxide, that would block electron flow from complex 4 to oxygen. So these are the structures of these compounds. I think it's interesting that there is really nothing that that is familiar or is not familiar, common about each of these structures. So in any case, I wanted to let you know what these compounds look like. So we can use a water pipes analogy of electron transport. So say I have these water pipes and I have a tap with water. So water will flow freely from the first pipe where it enters through all of the pipes and finally the exit, as long as none of the pipes are blocked. But suppose I put some kind of valve and I block the pipes. So what happens is as the water is added to the pipes will fill up. I've blocked the free flowing water. But what I could do is to get around that block, I could add, if we're using the water pipes analogy, I could just move this tap over to this pipe right here, and then I could have water flowing once again. That would keep these pipes full, but I would at least be able to continue the flow of water. In the cell culture, what you could do is you could add succinate, and succinate donates electrons to complex 2, so that's beyond the block. So as long as you had a nice supply of succinate, you could rescue your cells and they could continue on the pathway of electron transport. It would mean that all of the carriers ahead of the block would be reduced, but you'd at least be able to, as long as you had a nice supply of succinate, you'd be able to continue the electron transport chain. So what's happening here is if you added NADH and if rotenone were present, the electrons would not be able to be passed through complex 1 to coenzyme Q. But again, if you added succinate, succinate donates its electrons to FAD to form FADH2. And then FADH2 gives its electrons to coenzyme Q. So that bypasses this block. Now suppose we have antimycin A, we could do something similar in that we could move, if you use the water pipes analogy, we could move the pipes over here to cytochrome C. In the cells, what we would do is we could add ascorbate. That's an artificial substrate. But ascorbate would donate its electrons to cytochrome C, which again is past this block. So again, if you had an antimycin A block, electrons wouldn't be able to be passed from cytochrome B to cytochrome C1. But if you added ascorbate, you could rescue your cells and the electrons would be passed down the chain below the block to oxygen to form water, and you could continue the chain. Again, if the water pipes are full, and to continue the analogy, all of the carriers ahead of the block would be reduced, but at least you'd be able to continue the flow if you added an artificial substrate that donates electrons past the block. That is not the case for something like cyanide or azide or carbon monoxide. So here, the block is in complex 4 and the electrons aren't passed to oxygen. So here we can't bypass the block because it's the last step. Oxygen is the final electron acceptor in electron transport. And it does explain why carbon monoxide, azide, and cyanide are such deadly poisons. These things do other things to the cell as well, but this is one of the things that they do. Okay, so oxidative phosphorylation is a term that we use that describes how ATP is synthesized, and it is synthesized using the energy of electron transport. So to summarize, electrons collected from metabolic fuels, we started with glucose, but we can, other substances can feed into pyruvate and to acetyl-CoA as well. So they're Electrons have been stripped from these metabolic fuels. They were donated to the coenzymes. The coenzymes have passed those electrons and protons down the electron transport chain to oxygen to form water. So that describes electron transport. The electron transport chain has a free energy associated with that. We could just dissipate it as heat, or we could capture some of that energy in a useful form and use that to synthesize ATP. And this is referred to as oxidative phosphorylation. So Mitchell's chemiosmotic theory explains how that occurs. So what happens is that he proposed, and the experimental evidence shows that he is correct, and he in fact won a Nobel Prize for this work, that while electron transport is happening, that protons are pumped from the matrix to the inner membrane space, and that establishes an electrochemical gradient across the inner mitochondrial membrane. Electric, because the protons are positively charged, and chemical, because we have a higher concentration of protons in the inner membrane space than we do in the matrix. So remember that complexes 1, 3, and 4 participate in proton pumping. So that shows this here. So this is a nice summary of chemiosmotic theory. So if we look at these complexes here, complexes 1, 3, and 4. So notice that these pink arrows show that in complex 1, while electron transport is occurring, 4 protons are pumped in complex 1, 4 protons are pumped in complex 3, and 2 protons are pumped in complex 4. So that's a total of 10 protons per NADH that donates its protons and electrons to complex 1 in electron transport. So the electron flow, which is shown in this diagram in blue, so here if we follow the blue, that's the path of NADH. We go through complex 1, just kind of skip over complex 2 to Q, to C, to cytochrome C, to complex 4 to oxygen to form water. And of course the FAD takes the path through complex 2 to Q, and then the pathway is the same. So the electron flow is accompanied by this proton transfer, this proton pumping. So we get a chemical gradient that we can describe by the change in pH, and an electrical gradient that we can describe by delta psi. This works because the inner mitochondrial membrane is largely impermeable to protons. What happens to those protons then is these protons will come through this F sub 0 channel right here. So this is a transmembrane domain of the ATP synthase enzyme. And I have some videos that I can show you in a minute that show how the protons will stimulate the F sub 1 portion of this enzyme to couple ADP and phosphate to form ATP. So again, just to remind ourselves, complexes 1, 3, and 4 participate in proton pumping. Protons are moved or pumped from the matrix to the inner membrane space. That results in a deficit of protons in the matrix and an increase of protons in the inner membrane space. So we refer to this as the proton motive force, and it's the energy reservoir that drives ATP synthesis. So the gradient is both electrical and chemical. So this is an imbalance of protons. It's a non-equilibrium state. It does have an associated free energy that we can calculate, and we can think of the free energy associated with the proton motive force is the energy that we would have to put into the system to restore it to equilibrium. So we could calculate this, the free energy, if we consider the chemical and the electrical components separately. So this equation right here, we would enter the proton concentrations out. So this would be the proton concentration in the inner membrane space. This would be in. This would be the proton concentration in the matrix. And we have a difference. We have a greater concentration of protons in the inner membrane space and a lower concentration in the matrix. This equation describes the chemical portion of the electrochemical gradient. And this portion right here, the delta G equals the Z times the Faraday times delta psi. Z is the charge on the proton, which is +1, and delta psi is the membrane potential, which can be measured experimentally, and F is the Faraday, which is a constant. So if we put these together, we can calculate the free energy change associated with the proton motive force, or the electrochemical gradient that occurs across the inner mitochondrial membrane when electron transport is occurring. So it is a combination of chemical and electrical effects. So if we look at this picture right here, protons are pumped out of the matrix and to the inner membrane space. So this N side just means negative. So we have a negative charge on this side, and this is the P side, or the positively charged side, in the inner membrane space. And it turns out, if you do these delta G calculations, that the passage of 10 protons back into the matrix generates a proton motive force of about negative 200 kilojoules per mole. So the 10 protons is significant because if we think of one NADH, it transfers its protons and electrons to complex 1, which pumps 4 protons, complex 3, which pumps 4, and complex 4, which pumps 2. So 4 plus 4 plus 2 equals 10. So one NADH, reduced NADH, is responsible for the passage of 10 protons from the matrix into the inner membrane space, and that generates a proton motive force of about negative 200 kilojoules per mole. That corresponds perfectly with the calculation that we did earlier in the class when we considered the E values. We calculated the E value associated with the reaction when NADH donates protons and electrons to oxygen to form water. And if we do this calculation right here with the 1.130 volts being the delta E0 primed of that reaction, and this is the Faraday, and 2 moles of electrons are being transferred, we get about the same value. So that delta G0 primed is negative 220 kilojoules per mole. So what this means is that when NADH and H plus are passing protons and electrons down the electron transport chain to oxygen to form water, there's a free energy release associated with that reaction. That free energy is largely captured in the electrochemical gradient across the inner mitochondrial membrane that occurs during electron transport when complexes 1, 3, and 4 participate in the proton pumping. So again, this diagram doesn't show the number of protons, but complexes 1, 3, and 4 pump 4, 4, and 2 protons, respectively. So this shows the NADH donating its electrons and protons, and we're going down the electron transport chain eventually to oxygen to form water. So the pumping occurs. So again, 1 NADH is responsible for 10 protons being pumped from the matrix to the inner membrane space. Those protons will come across this F sub 0 channel right here and stimulate the ATP synthase to couple ADP and phosphate to form ATP. So approximately 1 ATP is produced for each complex that participates in the proton pumping. I'm going to refine that later on in the lecture, but for now, let's think of it as 1 NADH results in these 3 complexes participating in proton pumping, and that would yield 3 ATPs. Like I said, I'm going to refine that later, but let's just think about it as 3 ATPs for now. And we can look at this if we look at the energy drops that occur. So this is the redox potential over here on the left side, and this shows that these complexes that participate in electron transport, that the electrons are passed. Again, look at the bluey arrows here to follow the path. So the electrons are passed through these complexes, and the complexes are arranged in terms of increasing electron affinity to finally to get oxygen, in which oxygen has the greatest electron affinity. And if we look at these redox potentials right here, and if we use the equation that I just showed you earlier to calculate the delta G, that we find that between complex 1, complex 3, and complex 4, there are these free energy changes that, that occur. This is free energy change that is free energy that's released as these electrons are passed through complexes 1, 3, and 4. And we notice that sufficient energy is released to synthesize about 1 ATP. So we have at least the amount of energy we need, and in some cases we have more. But we have, in all of these three cases, we have enough energy to synthesize an ATP. So again, approximately 1 NADH is responsible for the synthesis of 3 ATPs. Notice for complex 2, the energy drop is not sufficient enough for us to be able to synthesize ATP, and notice that in complex 2 we have no proton pumping. So the way that ATP synthase works, so this is a simple animation here. So here's our drawing of the mitochondria, and we have the outer and the inner membrane, and we have our complexes that participate in proton pumping. So the animation showed that as the electrons and protons are passed down the electron transport chain to oxygen to form water, proton pumping occurs, and those protons, it's labeled here as complex 5, but I prefer the name ATP synthase since that's what it does. These protons will come through this F0 channel and stimulate the active site of the synthase to couple ADP and phosphate to form ATP. So let's look at the ATP synthase in some greater detail. So again, just to orient ourselves, these are pictures taken from several textbooks. So here's the inner mitochondrial membrane, and the portion that of the enzyme that resides in the inner mitochondrial membrane is considered to be F0, and the F1 is this portion of the enzyme right here. So notice that we have 6 subunits. We have 3 alphas and 3 betas. The active site for the synthesis of ATP is largely located on the beta subunit. We can actually see this in the electron microscope where these look like lollipops right here, where you can see the F1 domain here. And over here on the right, we can see a little bit more of the structural detail of the enzyme. So in the last slide, you could see that we had 3 beta subunits. So in this diagram, we have 3 beta subunits. The beta subunits can assume one of 3 conformations: loose, tight, and open. And at any one given time, 3 of the subunits are in one of those configurations. So you never have all loose or all tight or all open or 2 of one or 1 of the other. Loose and tight and open is all we have. The progression is loose to tight to open. So what happens is if we follow this subunit up here, so ADP and phosphate bind to the open configuration. Where it says energy right here, this is the energy of electron transport. This is the protons being pumped through from the matrix to the inner membrane space, and then coming back through the ATP synthase. So when this occurs, open goes to loose, loose goes to tight, and tight goes to open. So the progression is loose to tight to open, and then open goes back to loose. So in the open configuration, ADP and inorganic phosphate bind. Then we go from open to loose. They're still bound. And there's not another picture drawn right here, but we would go from open to loose to tight. And when this subunit goes to tight, then ATP is synthesized. So we can look at that over here. So if I look down here, ADP and phosphate are bound to the loose configuration. When loose goes to tight, then ATP is synthesized from ADP and phosphate. And if I look over here, this is a tight where the ATP was already synthesized. And when tight goes to open, then the ATP is released. So again, if we're thinking of integers, if we have 1 NADH, 3 complexes participating in the proton pumping, 3 subunits, then 1 NADH is responsible for 10 protons, and 10 protons leads to 1 complete rotation of the ATP synthase. So 1 NADH gives us 3 ATPs. I do have an animation right here that doesn't show the complete rotation, but it does show a little bit of what happens. So if we start here, So again, we have loose, tight, and open. This is a gamma subunit right here. So the gamma subunit is responsible for the rotation that occurs when the protons bind and is responsible for the change in conformation between the beta subunits. So ADP and phosphate bind to the loose configuration. And then that's the energy of electron transport. So notice that loose went to tight, and tight went to open, and open went to loose. So now that that this subunit right here is in the tight configuration, ADP and phosphate can be coupled to form ATP. Over here, this this subunit was already in the tight configuration and the ATP was already synthesized, so that can leave. And that's going to be shown here in the next frame. So the ATP leaves, and then this was in the tight configuration. So the tight configuration coupled the ADP and the phosphate. So then in the next rotation, which this video doesn't show, this will go to open, and then that ATP can leave. So this slide right here shows the a bit of the mechanism. So again, if we look at this this figure right here, ADP and phosphate will be used to, will be coupled to form ATP, and water is also a product because it's a condensation reaction. So if we look over here on the right, the beta subunits are shown in gray. The alpha subunits are shown in green. There is a little bit of the participation of the alpha subunit. We have an arginine right here. So if you look over here, you can see that the arginine, there's another arginine here, and a lysine here, and a magnesium ion. So all of these side chains perfectly position the phosphate so that a nucleophilic attack can occur of this beta phosphate right here. So ADP can gain this third phosphate to form ATP, and then that's the water that's going to be the leaving group. So I would imagine that there's some nice ion pairs here because arginine and lysine and magnesium are all positively charged, and the phosphate is negatively charged. So that would help stabilize the substrates when they bind to the enzyme, orient them properly, and the transition state isn't shown, but likely the side chains will stabilize the transition state because all enzymes have a role in transition state stabilization. So this is the tight conformation that's shown right here. So ADP and phosphate are used to make ADP when the beta subunit is in the tight conformation. So the way that ATP synthase works is that in order for this conformation to occur, the protons have to bind. And that is shown in this picture, but it's probably shown more effectively in some of the videos that I have here. So I will go to this video here. And
the membranes of mitochondria have functionally distinct regions with different protein compositions. The inner membrane is highly folded to form invaginations known as cristae. The inner boundary membrane and the crista membrane contain different proteins. The crista membrane contains the protein complexes of the electron transport chain, along with the ATP synthase enzyme that produces most of the cell's ATP. In this series of cryo-electron micrographs of a small yeast mitochondrion, we see long rows of ATP synthase proteins, indicated by the arrowheads, projecting like lollipops into the mitochondrial matrix. When the series of images is combined to create a three-dimensional model, we see that the ATP synthase proteins are arranged in double rows along a highly curved membrane. The individual ATP synthase molecules in each row form a dimer with a companion ATP synthase molecule in the adjacent row. The rows of dimers induce or stabilize these regions of high membrane curvature, which would otherwise be energetically unfavorable. Further refinement of such images has allowed the detailed structure of each dimer to be ascertained. When the electron transport chain is operating, it pumps protons from the matrix to the crista space. The structure of the cristae, with rows of ATP synthase dimers along each edge, helps to explain how the proton gradient is harnessed to make ATP. Protons pumped into the crystospace are thought to diffuse very rapidly along the membrane surface, with the ATP synthase rows creating a proton sink at the crystal tips. The protons flow down their gradient into the matrix through the ATP synthase enzymes, which convert the mechanical force into the chemical energy of a phosphate bond, joining ADP and phosphate into ATP. Thus, the cristae seem to work as proton traps that enable the ATP synthase to make efficient use of the protons pumped out of the mitochondrial matrix. It is interesting to note that this elaborate arrangement of membrane protein complexes is absent in chloroplasts, where the proton gradient is much steeper. With the cristae operating as proton traps, the protons flow rapidly through ATP synthase. Its rotor spins at about 8,000 revolutions per minute, generating 3 molecules of ATP per turn, or roughly 400 molecules of ATP per second.
So that would be a phosphoanhydride bond rather than the phosphate bond, as she says in the video. But I think you can see there how the protons are harnessed to rotate these C subunits right here, and that turns this gamma subunit, and that results in the change in the conformation of the beta subunits. So that's shown in this video here. So we can have a little bit more detailed view.
ATP synthase is a molecular machine that works like a turbine to convert the energy stored in a proton gradient into chemical energy stored in the bond energy of ATP. The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only protonated subunits can then rotate into the membrane, away from the static channel assembly. Once the protonated subunits have completed an almost full circle and have returned to the static subunits, an exit channel allows them to leave to the other side of the membrane. In this way, the energy stored in the proton gradient is converted into mechanical rotational energy. The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase, which catalyzes the formation of ATP. The F1 ATPase portion of ATP synthase has been crystallized. Its molecular structure shows that the position of the central shaft influences the conformation and arrangement of the surrounding subunits. It is these changes that drive the synthesis of ATP from ADP. In this animated model, different
tight,
conformational states are lined up as a temporal sequence
and then
as
open.
they would occur during rotation of the central shaft.
So loose, tight, and open. So you can follow each one of these subunits from loose to tight to open as you watch the video.
ATP synthase can work in either direction. If the concentration of ATP is high and the proton gradient low, ATP synthase will run in reverse, hydrolyzing ATP as it pumps protons across the membrane. To show the rotation of the central shaft, a short fluorescent actin filament was experimentally attached to it. Single filaments attached to single F1 ATPases can be visualized in the microscope. When ATP is added, the filament starts spinning, directly demonstrating the mechanical properties of this remarkable molecular machine.
So I think both of those videos show pretty powerfully how the protons that are pumped by complexes 1, 3, and 4 come back in through the F sub 0 portion of the ATP synthase, and then how it drives the rotation of the F sub 1 portion of the enzyme. And again, a full rotation results in all 3 of the subunits being able to synthesize each one synthesizes 1 ATP, so a full rotation will give you 3 ATPs. So all 3 beta subunits go from loose to tight to open, and when you're tight, you synthesize an ATP. So again, during electron transport, when all 3 complexes are engaging in proton pumping, you'll get a full rotation. So the proton gradient is absolutely necessary. So if there's no proton gradient, there's no free energy to drive the rotation of that gamma subunit, and you don't get those beta subunit conformational changes that are necessary for ATP synthesis. So we can introduce a concept called the P-to-O ratio. That's described as a relationship between respiration and ATP synthesis. So the P refers to the synthesis of a phosphoanhydride bond. So when you couple ADP and phosphate, you get ATP, and a phosphoanhydride bond is synthesized. So that's the P part of this. The O is the atom of oxygen. So if you look at the equations, and we show 1/2 O2. So that's an atom of oxygen. So the P-to-O ratio is the amount of ATP synthesized per atom of oxygen reduced. So the way I've been presenting this so far is that if one NADH goes through electron transport, and if you translocate 10 protons into the intermembrane space for all 3 of those complexes that are participating in the pumping, and if that gives you 1 complete rotation of gamma through the 3 ATP synthesizing units of F sub 1, that should give you a P-to-O ratio about 3. So that means that 1 NADH would give you 3 ATPs. For QH2, remember, we don't go through complex 1. We go through complex 2, which doesn't pump, and complexes 3 and 4. So that's 4 plus 2, that's 6 protons. That gives us a 2/3 rotation. So that means that 2 of the beta subunits are in the tight configuration, and you get 2 ATPs per 1 QH2. So that would give you a P-to-O ratio of about 2. It turns out, though, even though it's very satisfying to think of those P-to-O ratios as integers, 3 complexes participating in pumping, 3 beta subunits in a complete rotation, each giving us an ATP. There's a little bit of proton leakage in the inner mitochondrial membrane, so recent research has refined this. So we're going to use the P-to-O ratio of 2.5 instead of 3 for NADH, and 1.5 instead of QH2. And this diagram shows this fairly well. So what you should do now is you should pause the presentation. You should calculate this for yourself, and I'll show you the answer in the next slide. So if each NADH produces 2.5 ATPs, then you just would multiply 2 and 2 and 6 times 2.5 to get the amounts of ATP that each of those NADHs would synthesize. And here we have FADH2. Remember, that donates its protons and electrons to Q. So you would multiply this by 1.5 to get the amount of ATPs produced by coenzyme Q. Remember, we have some ATPs too. In glycolysis, we get 2 ATPs. For 1 turn of the citric acid cycle, we get 1 GTP. So we get 2 turns per mole of glucose. So that gives us 2 GTPs, and GTPs are energetically equivalent to ATP. So pause the presentation now, and then come back and see if your values agree with what I'm going to show you in the next slide. Okay, so again, if we multiply 2 by 2.5, we get 5 ATPs here. This is the 2 that we got from substrate level phosphorylation. Another 5 here, 15 here. This is 2 times 1.5, and we get 3 here. Again, substrate level phosphorylation in the citric acid cycle. And if you add these up, remember earlier we talked about the malate aspartate shuttle, where the two NADHs from glycolysis transfer their protons and electrons without any loss of energy to the mitochondrial matrix. So if you add up the ATP from oxidative phosphorylation and compare that to substrate level phosphorylation, you get 32 ATPs, but 4 of them are for substrate level phosphorylation and 28 are from oxidative phosphorylation. So having oxygen is a great advantage in terms of getting ATP out of your glucose. If you use the glycerol phosphate shuttle, remember that did occur with a loss of free energy. The protons and electrons from NADH in that shuttle gave their protons and electrons to FAD. So here we would take this and we would multiply by 1.5 instead of 2.5. Here again, though, we get 26 ATP from oxidative phosphorylation and 4 from substrate level phosphorylation. So we get 30 ATP. So we get a whole lot more ATP from the same amount of glucose if we oxidize it aerobically than anaerobically. A couple other things to note. One is that if we look at this reaction down here, remember we're going in the reverse direction. So we're taking ADP and phosphate and protons and we're synthesizing ATP. So what that means is we need to get our reactants into the matrix and then we need to export our ATP product out because the ATP is being generated in the mitochondrial matrix, but there are other parts of the cell where ATP is needed. So this is our ATP synthase right here. So that shows the protons coming in and using phosphate and protons and ADP to make ATP. So that means the ADP has to come in and the ATP has to come out. So this occurs via this adenine nucleotide translocase. It's an antiporter because we have ADP coming in and ATP coming out. We also have a phosphate translocase. This is a symporter because it brings a proton and a phosphate across in the same direction. So using the combination of all of these strategies, reactants are able to enter the matrix and the products are able to leave. So the evidence that supports this chemiosmotic hypothesis are these observations which can be made in the laboratory. So the pH of the intermembrane space can be measured. It's lower than the pH of the matrix. I'm not exactly sure how you would do this, but you can in the lab artificially impose a pH gradient. And if you do that, and if you give your cells in culture the raw materials, you will get ATP synthesis. You absolutely do need a closed compartment because if you're going to have that proton motive force across the membrane, you can't have a leaky membrane. The other thing is that compounds called uncouplers will dissipate the gradient. So that's the next thing I want to talk about. A classic DNP uncoupler is DNP is dinitrophenol, and it dissipates the proton gradient. And the way it does this is by uncoupling electron transport from oxidative phosphorylation. So electron transport still occurs, and there's a free energy associated with that. So if we are not capturing the free energy of electron transport to synthesize ATP, the energy is lost as heat instead. So if you look at the structure here, you might imagine that this molecule is sufficiently nonpolar to go across the membrane, and it is. But you might think that when a proton is lost and you have a negative charge, you might think that maybe the dinitrophenolate anion couldn't go across the membrane because of its charge. The thing is, it can actually, and the reason for that is that the dinitrophenolate anion is resonance stabilized. So we can draw these resonance stabilized structures down here. And you can see what happens is that the charges are delocalized. So that means that the dinitrophenolate anion can, in fact, cross a membrane because of the delocalization of the charges because of these resonance contributors to its structure. So this is how it works. So if we orient ourselves, so here we are in the intermembrane space, and all of the proton pumping complexes 1, 3, and 4 have pumped protons into the intermembrane space. So that means that in the intermembrane space, in the presence of all of that pumping, which occurs during electron transport, the pH is low and we have a high proton concentration. So that means if we added DNP to our cells in culture, then the DNP is likely to be protonated. So what happens is this is sufficiently lipid soluble that it can cross the membrane. So now here it is in the matrix, and the matrix has a higher pH and a lower proton concentration. That's going to favor the release of this proton to form the diphenylate anion. And again, that can cross through the membrane too because of its the delocalization of the charge. So that means that can come back. And once it comes back into the intermembrane space, it can become protonated again. So we have this cycle where the dinitrophenol that's protonated comes through the membrane, loses a proton, and then comes back. And it's constantly shuttling the protons that are pumped out of the matrix into the inner membrane space by complexes 1, 3, and 4. The dinitrophenol brings them back. And if the dinitrophenol is bringing them back, the protons aren't able to go through this F sub 0 channel, and you don't get any synthesis of ATP. So the dinitrophenol essentially collapses the proton gradient. So energy is released as electron transport, because electron transport is still happening. You still have oxygen and it's still accepting those protons and electrons, but since you're not capturing the free energy in the synthesis of ATP, the energy has to go somewhere and it's released as heat. So again, the reason this can occur is because of the delocalization of the charge that occurs on the dinitrophenolate anion. So in a lot of these pathways, we spend a lot of time talking about how these pathways are controlled. Normally in resting individuals, so if we're sitting in our chairs in front of our computers now, hydrolysis of ATP to ADP is low, so the concentration of ADP in our cells is not particularly high. But if we get up and run around, then our ATP will be hydrolyzed to ADP, and the concentration of ADP will go up, and that ADP increases the rate of electron transport. So we can go in the lab and we can show this, that oxygen consumption goes up. Oxygen consumption is a really good way to measure the rate of electron transport because oxygen is the final electron acceptor in electron transport. So if we looked at our cells and we had sort of a low amount of electron transport as measured by the low concentration of oxygen consumed, and if then we just added ADP to our culture, you can see that the slope of this line increases quite dramatically. So electron transport and oxidative phosphorylation will ramp up, and eventually all your ADP can be converted to ATP. So you'll exhaust the supply of ADP, and then you'll just continuing at the resting rate. So so this shows very dramatically how the ADP concentration can affect electron transport. So what that means is that we have to coordinate glycolysis and the citric acid cycle with the control electron transport, because glycolysis and citric acid cycle are supplying the electron transport chain with NADH and FADH2. So high concentrations of NADH and FADH2, which would occur in the presence of high rates of glycolysis and the electron transport chain, will fuel electron transport. Fatty acid breakdown, as we're going to see on Friday, also produces lots of acetyl-CoA that goes through electron transport. That gives us a lot of NADH and FADH2, so that will also fuel electron transport. So I will leave you with this this figure right here. There's a link to the animated figure which you can do on your own. There's a nice BioVision's powering the cell, where you can see the electron transport chain and the ATP synthase in action. But this is a nice summary right here. So all of these weeks that we've been going through glycolysis and the citric acid cycle. So these are all of the metabolites that will regulate the enzymes of these pathways. And here we're emphasizing the ratio of NADH. Again, so when the ratio of NADH and NADP plus is high, you can regulate your pathways this way. And again, because these pathways produce NADH, which fuel electron transport, everything has to be connected and regulated as a whole. These videos are also on Sakai, so I will lead you to this. But this is a really nice summary, I think. It shows pyruvate coming through the mitochondrial matrix and the citric acid cycle, the NADH and the FAD that is generated, the electron transport chain here where the NADH and the FADH2 feed in, the proton pumping that occurs, the protons that come back through the ATP synthase, the ATP that is made, and then these are the transporters over here. So I think this is a good summary of all the concepts that we talked about today. So that is it for today. We have already played the electron transport game. Normally, if things were normal, we would play it after we've learned all of this, but things are not normal. So here are my references. I hope this is helpful, and I will see you next time.